All right, let's dive into the world of receptor regulation.
It's fascinating, isn't it?
Yeah, these receptors on our cells, they're constantly adapting to signals, kind of like a volume knob.
Right, like adjusting how sensitive our cells are to different signals.
And it all ties back to keeping our body in balance, right?
Exactly, homeostasis. Too many signals and the cell gets overwhelmed.
Too few, and it misses important information.
Receptor regulation helps strike that balance.
Okay, so what exactly are receptors? Why are they so important?
Think of your cells as cities, with receptors acting as gates.
Gates that control what goes in and out. Makes sense.
Each gate has a specific shape, like a lock, waiting for the right key.
And those keys are called?
Ligands. When a ligand binds to its matching receptor, it triggers a response inside the cell.
So finding the right key for the right lock. But you mentioned adaptation. What does that look like?
Well, one way is through desensitization, hitting the brakes on a receptor's activity.
Like if it's being bombarded with signals, it gets tired. Less responsive.
Precisely.
How does that braking actually happen? What's the mechanism?
It involves some teamwork, actually. Enzymes called GRKs, G-protein-coupled receptor kinases.
GRKs. Okay, what do they do?
Think of them as taggers. They attach a chemical tag, a phosphate group, to the overstimulated receptor, a signal saying, time for a break.
So the GRK tags the receptor, then what?
Another molecule steps in, arrestin.
Arrestin, all right. What does that do?
They're like the tacklers, binding to those tag receptors, preventing them from sending more signals, like a temporary lock on that gate.
Wow, so GRKs tag and arrestins shut it down. Desensitization in a nutshell.
Precisely.
But what about when the cell needs to be more sensitive to a signal?
That's where upregulation comes in. The cell increases the number of receptors on its surface.
So more gates to let in more of a specific molecule? Interesting.
Makes it more responsive to the signal.
What might trigger that increase, that upregulation?
One common trigger is chronic antagonist exposure.
Antagonist.
Molecules that block receptors, preventing activation. Block a receptor for too long, the cell compensates.
By making more of that receptor, it's trying to listen harder.
Exactly, maintaining that balance, that homeostasis.
But what about the flip side? Decreasing sensitivity.
That's downregulation. The cell decreases the number of receptors, reduces sensitivity.
Like breaking up some of those gates to limit the flow?
A good analogy.
What causes downregulation?
Often, it's chronic agonist exposure.
Agonists.
Molecules that activate receptors, like turning a key in the lock, constant activation, and the cell might downregulate. Too much traffic through a gate.
And the city closes it off to ease congestion. Makes sense. Yep. So it's all about regulating that flow of information.
Precisely. And this upregulation and downregulation, it can happen in two main places.
Okay, so we have the cell surface.
Right.
And inside the cell itself.
Two main locations.
So desensitization with GRKs and arrestins, that happens on the surface.
Correct.
What about downregulation? Where does that take place?
Downregulation often involves a more complex process. It happens inside the cell, like a cellular recycling program.
Bringing receptors inside to be broken down or recycled.
Yeah.
A cellular ballet.
Constantly moving between the surface and the interior. A lot going on.
All this intricate action. But it's not just theoretical, right? This affects how our bodies react to medicines.
Absolutely. Real-world consequences.
Drug tolerance. Needing higher doses over time to get the same effect.
That's often a direct result of downregulation.
So your body tries to maintain balance even in the presence of medication.
Precisely. That's why understanding receptor regulation is crucial for drug development.
If we can figure out how to minimize downregulation or even use upregulation,
we could create better treatments, fewer side effects, potentially.
This is fascinating. It sounds like a whole world of research going on here. But how do scientists actually study these tiny receptors, track these changes? It seems so difficult.
It is a challenge, but scientists have developed some ingenious techniques.
Like what? How do they peer into this microscopic world?
Well, one classic technique is called radioligand binding assays.
Radioligand binding assays.
Attaching a tiny tracking device to those molecular keys we talked about, the ligands.
Using radioactive versions of those ligands.
To see how they bind to specific receptors. Measure receptor number.
Ah, like a cellular census. Counting the receptors and how they're distributed. But that's just one piece. We want to see those receptors in action, right?
Of course. And we can, using techniques like fluorescent labeling, attaching glowing tags to receptors.
Giving them tiny glow sticks to track their movement under a microscope.
Exactly. We can see them clustering, moving apart, even going inside the cell for recycling.
Incredible.
It is a dynamic view of this intricate dance.
Okay, we can count them? track their movements. Can we manipulate them, though? Change their expression levels, their genetic code?
That's where genetic manipulation comes in. Tools like CRISPR-Cas9,
CRISPR.
we can edit genes, including those that code for receptors, like a molecular toolkit to tweak those cellular gatekeepers.
So we can create cells with more receptors, fewer, or even with altered functions.
Opening up a whole new world for research.
Okay, I think I'm starting to understand this receptor regulation, this dynamic process of adjusting sensitivity, changing the number or activity of receptors.
Precisely.
And all this is driven by a variety of factors: type of signal, duration of exposure, even the cell's internal state.
It's a complex system constantly evolving.
Okay, so we've talked about desensitization, upregulation, downregulation, how these processes can happen on the surface or inside the cell. And we touched on how it all plays into drug tolerance. Anything else we should know about the implications of receptor regulation?
Well, one area where receptor regulation is becoming increasingly important is personalized medicine.
Personalized medicine. Intriguing. How does receptor regulation fit in?
Imagine treatments tailored to an individual's unique genetic makeup, their specific receptor profiles. We could analyze their genes to predict their sensitivity to certain drugs, their likelihood of developing tolerance.
Wow. So instead of a one-size-fits-all approach to medicine?
We could personalize treatments based on their individual receptor profile, a roadmap for navigating those treatments.
A roadmap. That's a great analogy. But it's constantly being refined, right, as we learn more.
Exactly. And we are learning that different subtypes of receptors can have slightly different roles and responses.
Subtypes? I thought a receptor was a receptor, like an on-off switch.
It's more nuanced than that. Even within a family of receptors, there can be variations, like different models of the same car.
They might look similar, but...
They have different features under the hood.
So how do these subtypes affect receptor regulation?
Well, let's consider dopamine receptors. They're involved in mood, motivation, movement, several subtypes, D1, D2, D3, and so on, each with slightly different structure and function, and they're located in different parts of the brain.
So drugs targeting specific dopamine receptor subtypes could have very different effects.
Precisely. Understanding the unique properties of each subtype is crucial for developing more precise treatments.
Okay, so let's get specific. Can you give us an example of how dopamine receptors are regulated in the brain?
Sure. Let's look at Parkinson's disease. It affects movement, caused by the loss of dopamine-producing neurons in a specific area of the brain, the substantia nigra.
So a shortage of dopamine disrupting movement.
Right, and we treat it with a drug called levodopa, a precursor to dopamine. It crosses the blood-brain barrier, converts to dopamine.
Helping to restore those dopamine levels.
Exactly. But here's where receptor regulation comes in. Continued use of levodopa can lead to downregulation of dopamine receptors.
The brain senses this constant influx of dopamine from the medication, tries to regain balance.
And that downregulation contributes to levodopa-induced dyskinesias, involuntary movements, a side effect of long-term treatment.
So you need the medication, but it triggers changes that lead to side effects.
A delicate balancing act. And it highlights how important it is to understand receptor regulation in drug development and treatment.
Okay, so that's dopamine receptors in the brain. What about other types of receptors? Any other interesting examples?
Of course. Receptor regulation is happening all over the body, in every organ system. Let's consider insulin receptors. They regulate blood sugar levels.
Insulin. That's the hormone that helps our cells absorb glucose from our blood.
That's right. When insulin binds to its receptor, it triggers a chain of events, leading to glucose uptake, lowering blood sugar.
So what happens with insulin receptors in people with type 2 diabetes?
In type 2 diabetes, the body develops insulin resistance.
The cells become less responsive to insulin.
Exactly. And one of the mechanisms behind that is downregulation of insulin receptors.
So the cells are constantly exposed to high levels of insulin. They try to dampen the response.
And that downregulation makes it harder for insulin to do its job, leading to further increases in blood sugar levels.
It's a vicious cycle. So receptor regulation is involved in a wide range of diseases.
It is, and understanding it is crucial for developing new treatments. For example, research into enhancing insulin receptor sensitivity, or preventing downregulation of dopamine receptors in Parkinson's.
Wow, this field could really revolutionize medicine.
It has that potential, and it's constantly evolving, new discoveries all the time.
We've covered a lot today. Receptor regulation, different types of receptors, desensitization, upregulation, downregulation, implications for drug tolerance and personalized medicine. Anything else we should touch on before moving on?
There's one more fascinating concept: Receptor crosstalk.
Receptor crosstalk.
The idea that different receptors can communicate with each other, influence each other's activity.
So it's not just individual receptors. They interact and influence each other.
Exactly. And this crosstalk can lead to synergistic effects. The combined activation of multiple receptors creates a response greater than the sum of its parts.
Like different instruments playing together.
A beautiful analogy. But that harmony can be disrupted by disease or drug interactions.
Creating a cacophony. So understanding receptor crosstalk is important for deciphering health and disease.
Precisely. It's a rapidly evolving field with the potential to revolutionize drug development and personalized medicine.
Now, how do scientists actually study receptor regulation? We touched on some techniques earlier. Can we delve into those a bit deeper?
Of course. One of the most powerful tools is radioligand binding assays.
Right, the radioligand binding assays.
It helps quantify the number of receptors in a tissue or cell preparation, like taking a census of those cellular gatekeepers.
Using radioactively labeled ligands to track how they bind.
Gives insights into receptor density and affinity, like attaching a tracker to those keys and watching them dock onto those cellular locks. And by comparing binding of different ligands,
We see their preferences, how they interact.
exactly, crucial for developing drugs that effectively target those receptors.
And for visualizing those receptors in the cell.
We have fluorescent labeling techniques, tagging receptors with glowing markers to track their movements within the cell.
Like tiny beacons, illuminating their activity.
And using different colors, we can track multiple receptors simultaneously, see how they cluster or separate in response to stimuli.
Like a choreographed dance, each receptor with a glowing costume.
Exactly, essential for understanding cellular communication and signaling.
Now, what about manipulating receptors directly? Changing their expression levels or even their genetic code?
That's where genetic manipulation techniques come in. Tools like CRISPR-Cas9, it's revolutionized gene editing, including for receptors.
CRISPR. Like a molecular scalpel.
We can introduce mutations, delete genes, study the functional consequences, like conducting an experiment, observing how the symphony changes when we modify or remove instruments.
And these experiments reveal the roles of specific receptors in health and disease.
Precisely. Unprecedented insights.
Now, there's also a newer technique, single molecule imaging.
A cutting-edge approach allows us to visualize individual receptors in real time. Incredible detail.
Like a microscopic camera capturing the solo performance.
Revealing new insights into the dynamics of receptor activation, desensitization, and trafficking.
The receptor's life story.
Helping us understand the molecular mechanisms governing cellular communication.
All these techniques are powerful. But they often study cells in isolation, outside their natural environment.
True. These in vitro studies are valuable, but they don't fully capture the complexity of regulation in living organisms.
How do we bridge that gap then?
That's where in vivo imaging techniques come in, visualizing receptors in living animals, observing their natural behavior,
Like taking those receptors on a field trip.
giving us a glimpse into the dynamic interplay between receptors, cells, and tissues.
So we can see how they function in the real world.
Exactly.
Amazing. So how does all this understanding of receptor regulation actually impact drug development? It seems pretty crucial.
It is crucial. We need to develop safe and effective medications. And as we've seen, drugs can alter receptor sensitivity,
Leading to problems like tolerance.
dependence, even unexpected side effects. It's not just about finding a drug that binds, it's about understanding how that binding will affect receptor behavior over time.
We need to think long-term.
Absolutely. If we consider receptor regulation early in development,
We can design better drugs.
more precise, less likely to cause tolerance, and fewer side effects. It's challenging, but the potential rewards are huge.
It's like a molecular chess game, trying to anticipate the cell's next move. Develop drugs that outsmart those adaptations.
A good way to put it. The more we understand receptor regulation, the closer we get to truly personalized medicine,
Treatments tailored to each person.
to their unique genetic makeup and receptor profile.
That's incredible. Imagine minimizing trial and error with medications, avoiding adverse reactions, maximizing benefits. That's the promise of personalized medicine.
Powered by a deep understanding of receptor regulation, it's an exciting field with so much potential.
So to wrap up our deep dive, receptor regulation is a dynamic and essential process. For virtually every aspect of biology, these gatekeepers on our cells are constantly adjusting, maintaining balance, keeping everything running smoothly.
And as we've learned, this cellular dance has profound implications. Understanding disease, developing new drugs, personalizing treatments, a field ripe with possibilities. Immense promise for the future of medicine.
Learning about this has given me a whole new appreciation for the complexity of our cells.
Mm.
So much happening at such a tiny level.
It is remarkable. And the more we learn, the more we realize how much we still don't know. Each receptor has its own story, its own partners, its own dance. A captivating field. I encourage you to keep exploring.
You've certainly inspired me to learn more. Thanks for taking us on this deep dive into receptor regulation. It's been eye-opening.
My pleasure. And remember, the world of science is full of wonders waiting to be uncovered. Keep those neurons firing and those receptors receptive.
It really is. So where do we go from here? What else is there to explore about receptor regulation?
Well, one area that's really intriguing is the idea of biased agonism.
Biased agonism. What's that?
So traditionally, we've thought of receptors as being like simple on-off switches. A ligand binds, the switch flips, and the signal is sent.
Right, like a key in a lock.
Exactly. But it turns out that some ligands, these biased agonists, can actually fine-tune the receptor's response.
Fine-tune. How so?
Instead of just turning the receptor on, they can activate specific signaling pathways while leaving others untouched. It's like having a dimmer switch instead of a simple on-off switch.
So different ligands can elicit different responses from the same receptor. That's amazing. What are the implications of that?
Well, it means we might be able to develop drugs that are even more precise, drugs that can target specific signaling pathways without activating unwanted ones. This could lead to therapies with fewer side effects and improved efficacy.
That's really exciting. It's like targeting a specific instrument in that cellular orchestra we talked about earlier. Instead of just turning up the volume on the entire symphony, we can focus on a particular melody.
Exactly. And this concept of biased agonism is opening up a whole new frontier in drug discovery.
This is all so fascinating. It really makes you appreciate the complexity of our cells and how much we still have to learn.
Absolutely. And the more we learn about receptor regulation, the more we realize how crucial it is for virtually every aspect of human health, from basic physiological processes to the development of new therapies for a wide range of diseases.
Well, on that note, maybe it's time to shift gears a bit and dive into some specific examples of how receptor regulation goes awry in disease.
Sounds good. Where do you want to start?
Well, we've talked about Parkinson's disease and type 2 diabetes, but what about other conditions, like addiction? How does receptor regulation play a role there?
Addiction is a complex disorder, but receptor regulation is definitely a key player. Let's take the example of opioid addiction.
Opioids like heroin or prescription painkillers.
Right. Opioids work by activating opioid receptors in the brain. These receptors are involved in pain relief, but they also play a role in reward and pleasure.
So when someone takes opioids, they experience a surge of dopamine in the reward pathway of the brain.
Exactly. And this dopamine rush reinforces the drug-taking behavior, making it more likely that the person will seek out opioids again.
But over time, the repeated use of opioids leads to changes in receptor regulation, right?
It does. The brain adapts to the constant presence of opioids by downregulating opioid receptors. This means that the person needs higher and higher doses of the drug to achieve the same effect.
So tolerance develops.
Yeah.
And what happens if the person suddenly stops taking opioids?
That's when withdrawal symptoms set in. The body is no longer receiving the artificial stimulation it's become accustomed to, and the downregulated receptors are struggling to function normally. This can lead to a range of unpleasant symptoms.
Like muscle aches, nausea, anxiety, insomnia.
Exactly. And in severe cases, withdrawal can even be life-threatening.
So receptor regulation plays a central role in both the development and the treatment of opioid addiction. It's a vicious cycle.
It is. And it highlights the importance of understanding how drugs interact with receptors and how those interactions can change over time.
Are there any other examples of diseases where receptor regulation is implicated?
Oh, there are countless examples. Receptor regulation is involved in everything from heart disease to cancer to autoimmune disorders. Let's take a look at heart failure, for instance.
Heart failure. That's a serious condition where the heart can't pump blood effectively.
Right. And one of the key players in heart failure is the beta-adrenergic receptor.
Beta-adrenergic receptor, what does that do?
It's a type of receptor that's activated by adrenaline and noradrenaline. These are hormones that are released in response to stress.
So when we're faced with a stressful situation, our heart rate and blood pressure increase.
Exactly. And the beta-adrenergic receptor plays a crucial role in this fight-or-flight response.
So what happens to these receptors in heart failure?
In the early stages of heart failure, the body tries to compensate for the weakened heart by increasing the activity of the sympathetic nervous system.
The sympathetic nervous system is responsible for that fight-or-flight response.
Right, so more adrenaline and noradrenaline are released, stimulating the beta-adrenergic receptors in the heart. This helps to increase heart rate and contractility, trying to maintain blood flow.
So the body is trying to rev up the engine, so to speak, to compensate for the failing pump. But what happens over time?
Over time, this chronic stimulation of beta-adrenergic receptors leads to downregulation. The heart becomes less responsive to those fight-or-flight signals.
So it's a case of too much of a good thing. The initial compensation mechanism ends up backfiring.
Exactly. And this downregulation of beta-adrenergic receptors contributes to the progression of heart failure. The heart becomes weaker and weaker, and eventually it can no longer keep up with the body's demands.
So receptor regulation plays a critical role in the downward spiral of heart failure. Are there any treatments that target these receptors?
There are. Beta blockers are a class of drugs that block the action of adrenaline and noradrenaline on beta-adrenergic receptors.
So they help to dampen that overactive sympathetic nervous system response.
Precisely. And by blocking these receptors, beta blockers can actually help to improve heart function and slow the progression of heart failure.
That's incredible. It's like taking the foot off the gas pedal and giving the heart a chance to rest and recover.
A great analogy. And it highlights the importance of understanding receptor regulation in the context of treating complex diseases like heart failure.
Okay, so we've seen how receptor regulation plays a role in addiction, heart failure, and a host of other conditions. It's really amazing to see how these tiny molecules can have such a profound impact on our health. But what about the future? Where is research in receptor regulation headed? What are some of the most exciting areas of exploration?
That's a great question. The field of receptor regulation is constantly evolving. New discoveries are being made all the time. One area that's particularly exciting is the development of allosteric modulators.
Allosteric modulators, what are those?
They're drugs that bind to a different site on the receptor than the endogenous ligand.
So not the same binding site as the natural key.
Right, they bind to a different spot, and by doing so, they can either enhance or inhibit the receptor's activity.
So they can fine-tune the receptor's response like those bias agonists we talked about earlier?
Exactly. But allosteric modulators offer even more flexibility and precision. They can selectively target specific receptor subtypes or even specific signaling pathways.
So it's like having a toolkit with a whole range of wrenches and screwdrivers, instead of just a single hammer. We can fine-tune those cellular machines with much greater precision.
Exactly. And this opens up a whole new realm of possibilities for drug development. We can potentially create therapies that are more targeted, more effective, and with fewer side effects.
That's incredible. It sounds like the future of medicine is personalized, precise, and powered by a deep understanding of receptor regulation.
I think you're right. And as we continue to unravel the complexities of this intricate cellular dance, we'll undoubtedly uncover even more amazing discoveries that will transform the way we treat disease.
This deep dive has been an amazing journey, from those tiny molecular gatekeepers to the grand symphony of cellular signaling.
It's been a pleasure exploring it with you. And remember, there's always more to discover, so keep those neurons firing and those receptors receptive.
It really is. So is there anything else about the future of this field that has you particularly excited?
Oh, absolutely. One area that I think holds immense potential is the intersection of receptor regulation and artificial intelligence.
AI and receptor regulation. How do those two fields come together?
Well, as we've discussed, receptor regulation is incredibly complex. There are so many factors involved, so many moving parts. It can be really challenging to make sense of all the data.
Right. Even with all the techniques we've talked about, it's still like trying to piece together a giant puzzle.
Exactly. But that's where AI can come in. Machine learning algorithms can analyze massive data sets, identify patterns, and make predictions that would be impossible for humans to do on their own.
So AI could help us to see the bigger picture in receptor regulation, to connect the dots in a way that we couldn't before.
Precisely. And this could have a huge impact on drug development. Imagine being able to use AI to predict how a new drug will affect receptor behavior in different individuals, or to identify new drug targets based on patterns in receptor regulation.
That would revolutionize the way we develop new therapies. It would be like having a crystal ball that could tell us which drugs are most likely to be safe and effective.
It's a powerful idea, and it's already starting to happen. There are research groups all over the world using AI to study receptor regulation and develop new drugs. It's a really exciting time to be in this field.
It sounds like it. So to sum it all up, Receptor regulation is this dynamic process of fine-tuning cellular sensitivity to signals. It plays a role in virtually every aspect of human health and disease.
And thanks to advances in technology, we're finally starting to unravel its complexities.
And with the help of AI, we may be on the cusp of a new era of personalized medicine, where treatments are tailored to an individual's unique receptor profile.
It's a future filled with possibilities, a future where we can harness the power of receptor regulation to treat diseases more effectively and improve human health in ways we can only imagine.
Well, I have to say, this deep dive into receptor regulation has been truly mind-blowing. It's incredible to think about all the intricate processes happening inside our cells every second of every day. Thank you so much for sharing your expertise and taking us on this journey.
It's been my pleasure. And remember, the world of science is full of wonders waiting to be uncovered. So keep exploring, keep asking questions, and never stop learning.
Great advice. And to our listeners, thank you for joining us on this deep dive. We hope you found it as fascinating as we did. Until
Okay.
next time, stay curious.